endemism in microbial communities is another really interesting, long-standing question, and how can we make headway on that problem? There's also lots of what I call programmatic questions. These are questions that funding agencies or companies might care about. In the U.S., certainly the Department of Energy is interested in answering questions about the carbon cycle. And where is the carbon cycle being influenced the most by microbial processes versus geochemical processes and so forth. And so we see opportunity, for example, in focusing sequencing efforts in the permafrost, or maybe in certain terrestrial ecosystems that have high turnover in carbon, or certainly in the ocean. And are there novel mechanisms that mediate the role of carbon pathways? We certainly are very interested in diversity relationships in the community, both from a fundamental standpoint. Can we understand what makes a community stable, and can we understand how you might engineer a community by swapping out members of that community? That's an interesting one, both for natural systems and for artificial systems. And searching for novel proteins or novel functions in the environment that might be relevant to energy research, bioremediation, or bioprocessing is another very applied kind of outcome that a project like this would inform, because it would build the world's largest database of diversity that you could then mine in various ways. We think that, that there's work that shows that maybe half of the living protoplasm on Earth is microbial. This means that microbes probably play a much more important role in climate processes and fundamental biosphere processes than the macroscopic community would actually like us to believe. And this is interesting because it means that the interest of the research agencies in taking a fresh look at the role of microbes in climate is starting to have support. And this is an area, for example, the Department of Energy in the U.S. is quite interested in. Can we make a strong connection between what's happening in the microbial world and in the interchange with the atmosphere and biosphere? One of the projects that we're trying to do at Argonne is to gain an improved understanding of how we might include microbial processes in climate models and actually treat them as first-class objects as opposed to as boundary conditions, which is done now. And constructing effective ways of representing metabolism of microbial communities. I won't go through this in detail, but there's mechanisms for going from metagenomics data to reconstructed organisms, to building models of those organisms' metabolism, to building a model of a community's metabolism that's predictive enough to be able to articulate what overall role that community might be playing in a particular environment. There's, of course, many other reasons for doing this. There's some recent work at Berkeley, for example, that showed that our understanding of how the microbial world uses metals is really just beginning to be understood. There's much more diversity in metals that are used in proteins for mediating or for driving reactions of various types that before were not known. And this was only coming from studying these systems in much greater detail. And there's still enormous interest in just biodiversity more generally. So we think that EMP is sort of at the right time. It's leveraging the availability of advances in sequencing. It's also coming at a time when sequencing centers are sort of changing their model. Some centers, like BGI, for example, have embraced scale, right? Have embraced having a capability that is unique, perhaps, on the planet. And this is a kind of project that really could gain great leverage from that kind of recognition of sequencing centers. At the same time, there's likely to be many sequencing centers, sequencing capabilities pushed out into very small labs. And if we can find a way to integrate standardized protocols, each of these individual labs could also make a contribution. We need this to be community driven, but it's got to be coordinated in some fashion. Fortunately, we don't have to invent things like Twitter and Facebook, right? They're already here. So we can do open real-time coordination and real-time data availability. And this was something that was just amazing to me last weekend on the E. coli strain, when there were a spontaneous set of people working on this, sharing data, sharing observations, sharing tools, and none of it had been coordinated ahead of time. And it made me think that, gee, if we just had a little bit of coordination, and there was an interesting science result that comes out of some early work, people will just flock to it. It's sort of flash mob science, right, is what we're talking about. And that's really the new way of doing things. And the younger generation, many of you here, may sort of get that instinctively. And that's what this project would have to do. Mention those. So what are some of the things that we would produce? Well, we would produce these metagenomics data sets from many, many thousands of environments with a standardized metadata. That's very easy to say. Well, it's a long sentence, but it's relatively easy to say, very hard to do. We have to have a georeference inventory of the global 16S sequences so that we can figure out what we've seen where, before, and when. We want to have reference genomes that could be recovered from these metagenomic sequences. In cases where we've got very deep coverage and the diversity distribution is right, we should be able to recover many nearly complete genomes. We also need reference genomes. A project like Nikos is leading to produce thousands of reference genomes that would allow us to interpret the metagenomes that we see. We would build community structure profiles for many thousands of communities, and these would be synchronized in time and correlated with the physical parameters so that you could understand, say, time evolution of these communities. And we also produce a microbial protein catalog that would capture global protein and gene diversity, sort of like the STAR catalog that the other guys produce. Big reference book of all the known proteins that have ever been seen on Earth, for example. So the way we thought about getting this going is through a pilot. And there's a couple of, one way to think about this, so this is really more of a proposal than anything else, is a pilot project that would try to simultaneously make headway on the technical issues, as well as highlighting and demonstrating some of the scientific value. And we might pick a handful of problems, say making headway in global microbial diversity, or trying to characterize global dispersal potential, or global carbon partitioning, some headway in that, or your favorite problem. Right? And the idea here would be to loosely cluster projects that are related to these questions into clusters. Okay, so say the global microbial diversity one would have a series of projects, and these projects might explore variation across environmental gradients, availability of light, pH, and so forth, and do this in a way that allows, in a very public, open way, right? So people can see what the various gradient studies would be, and then propose things that are orthogonal to what people are already doing. So you can imagine a cluster of projects in this area. You can imagine a cluster of projects trying to characterize global dispersion. This is the determining whether everything has at least the potential to be everywhere in terms of exposure, and what's in the details of these distributions, right? So this would involve deep sequencing, particularly phylotype type sequencing, 16S sequencing, in many, many places, and looking for what taxa are common, or what OTUs are common to places, and what are geographically distinct. Well, carbon would be another target. We find communities that are particularly relevant, like permafrost, and do a series of studies there, try to reconstruct the communities that mediate the form in which carbon is released when permafrost melts. Is it carbon dioxide? Is it methane? Is it internally captured through metabolic processes and put into biomass in other ways. And this would be temporal and spatial samples from around the world, different environments that are particularly interest to people working in the carbon cycle. What would tie these together in a pilot, again, is this effort to construct some standards. And we need to have a way to bootstrap this, so we try to choose some standards for DNA extraction. Again, it would be imperfect and maybe inadequate to start with, but we started some standard protocols for sequencing and some standard pipeline for informatics, and have put into place some process, some EMP, what we call the EMP process, again, very open process that would create a forum for experts to discuss the relative merits of the standards, try to build consensus around ones that we think would work, try them out, and have a feedback loop as to whether the community is happy with them or need to change them, what's preventing them from being adopted or whatever. So in some sort of simple-minded sense, you can imagine the EMP process is really defining these standards, and lots of individual projects that buy into these standards result in many, many data sets that now have the look and feel of a more coherent project. And so one way to think about this is that you've got all kinds of people out there in the community that are designing studies, hypothesis-driven studies, sampling, building their collections and so forth. And on the right-hand side, we imagine many downstream applications of the kinds of data that would come out of EMP. But what we have in the middle is this piece that really has to be high throughput and relatively standardized, right? Sample part sequencing and some quality mechanism. Lots of different analysis will be done, but we've got to have this middle part. It can be done in multiple places, but where people have confidence in it, so they're willing to actually sign up to this model as opposed to doing their own thing. And part of what we need to do in the pilot is debug what the middle looks like. The other thing we have to do is work out from the informatics and computing side, how would we handle an influx of data driven by a project like this. And here's a schema that Volker or Jack dreamed up last year. This is a schema as a starting point for what you might do to handle this. Of course, we would need to involve a large community in bootstrapping this. But it also could drive some development within the sequencing centers. This is something that I think we're interested in talking to BGI about. A project like the EMP